Thank you for being interested in ASAP. This video is a short tutorial on how to use ASAP. In this whole video, I will just use a dataset from Duke from 2015 that that actually contains 5 different cell types, 3 neuronal tissues, 1 heart, and 1 brown fat. So when you first access this asap.epfl.ch web page, then you see this screen where you have whether the ability to send your dataset through the sandbox. Then you can upload it, you can work with it, or you can use example dataset that we already uploaded in public dataset. Or if you want to keep tracks of your data, then you can just create login and create a new project. So here is the login page where you can whether sign up for a new account or enter your credentials. So that's what I will do. Now is your project space. So you can see all the projects that you created that are saved here. And you can see also the public projects that were made public by us or by other users. So here you can see the first project, which is not public, so it's only seeable by me. And if you want, so you can create a new project. So that's what I will do right now. So here I will upload my count matrix or UMI matrix. First I need to put a project name. So the data is from Duke, so I will put Duke 2015. It's mouse data, so you can select the organism. So this organism is mainly used for enrichment analysis. It's not very mandatory, but if you have it, if it's present in the list, then it's always better to have it for helping the enrichment. So these organisms are based on the ensemble database. So there is a full 69 species from the full ensemble database since release 47, I think. Then you can upload your dataset in the correct format. There is an example file if you want to check. And then when you hit the create project button, then it sends the data and creates the project. Here is the parsing result page. So you can see whether your data was correctly parsed, how many genes you have, how many cells, if your data is a count matrix, or if it's already normalized data, which can be actually important because some tools are only working if you have a count data. So if it's normalized, then you will only have a restricted set of tools. Then you can see how many of your reads map to protein coding genes, ribosomal or mitochondrial RNA, based on the ensemble data. And also if you have ERCC in your data, it's automatically detected. It needs to start with ERCC dash, the name of the gene. And then you can use it, for example, in the next step for normalizing your data. You can also attach a batch file, which is basically a mapping between your sample name and the number of the batch, if you want to do some batch effect correction. And later on, so it's not already available, but later on we will implement also the ability to upload more metadata and annotation of your samples. Then the next step is the filtering step. So, okay, now we have our data in ASAP. We want to first remove all the genes that are lowly expressed, or the genes that are not varying enough, so they are not really interesting for the further analysis. So there are many methods available on ASAP, you can see. So whether you don't want to filter at all your data, or you can use any of these methods. So what is actually interesting in ASAP is that you can actually filter, then go to the visualization pane and look at your data if you see the clusters that you're expecting to see. And if you don't see them, then you can go back to the filtering step, reset the filtering with maybe another method or a more stringent cutoff. And then you can start again until you actually see your clusters in the visualization pane. So it makes it very easy to filter your data in different ways. So here I will use a variance tool, which basically just keeps the most varying gene. So here I select the 5% most varying gene. But you can also select the most variable gene method from, well, actually uses the Deplancke et al. Method. So there's, again, many ways of doing this. So here you see the results of the filtering step. So you have a list of plots that were generated that shows, for example, the number of expressed genes per sample, or the cumulative number of expressed genes when you take the cells as bulk, which is very classic in single cell data. And then now that you have a good look at your original data, then we can go to the normalization step. So here again, in the normalization step, you have many tools available, SCLVM, etc. So here I focus on SCLVM because it contains many different options. So for example, the first one, you can actually perform batch effect correction using Combat. So this step is actually available in every of the normalization methods, but you need a batch file, as I said before. So if you actually edit the project, you can add a batch file in your project. And then this option will be available here. It's great because you cannot do it because there's no batch file available. Then you can, while you're normalizing, you can also keep only the most variable gene, again with this Deplancke et al. Method. And if you have ERCC detected, then you can also normalize using this ERCC spike-ins data. But here, for this data, I will use VOOM method from the Lima package because it's very easy to use and it's very fast also to execute. So all the tools are run on our server, so nothing is run on your computer. And again, when the normalization ends, then you have some reports like plots to see how the normalization actually normalize your gene and how the distribution of your gene expression is for each sample. And automatically, when the normalization is finished, the visualization plots starts to be produced. So you have actually 4 different. You have PCA, t-SNE, MDS, and ZIFA. You can see here actually that ZIFA is still running on our server, so it's not finished. But PCA, t-SNE, and MDS are already done, so you can already see the results, and you can continue working with your project. And when ZIFA will be done, then it will pop up as a green check. So first you have the results of the PCA. So you actually have 2 plots, the 2D and 3D plots, which shows respectively the 2 first principal components and the 3 first principal components of the PCA. So you can see clusters, you can see also the names of the cells, and you can rotate the 3D plot if you need to. Then you also have the t-SNE plots and MDS plots that are very similar to the PCA plots. So if we go back to the MDS plot, we can actually overlay colors based on, for example, the gene expression. So here I will select a gene that I know is expressed in brown adipocyte cells. That is one of the 5 cell types that is available in this dataset. So you type adipoq and then it's auto-completed. And here you can see the expression of adipoq on the different clusters, and it's more expressed in the BAT cells, which are the brown adipocyte cells. For better visualization, you can whether remove the cell names, add legends, remove legends, and you can also put the plot in full screen and still continue working with it. So now we have the correct visualization, we see all the clusters we want to see, so the next step is to do clustering. This step we have many methods available, k-mean, SPAM, SE3, and hierarchical clustering. So you can run any of these clustering on your original dataset, normalized dataset, or even on the t-SNE data and PCA data. And you can select the number of clusters, or you can also say you want an automatic recognition of the best number of clusters, and it will be done by a silhouette plot. So you see that the methods can be run in parallel actually, and once they are finished, then you have green boxes that appears. So those green boxes are the clusters that are found by the different methods. So you have 2 or 5 clusters here. And you can also have like additional plots, like the silhouette plots that you can download, and you can also download the clusters actually as a single file. Then if you click on the green boxes, then you can actually see the list of cells that belong to the cluster. And if you want, you can add them to something we call selection, which actually help you then to see those cells in particular in the visualization plots. And you can also use this selection for differential expression afterwards. So here I will create a selection based on the 2.1 cluster, and I will rename it CRD because this is a cluster that contains only CRD cells, which are cardiac cells. And now I go to the differential expression step, where again there are many methods available, and you can do differential expressions between whether two selections that you already made or one selection versus the rest of all the cells. Again, you can also select if you want to do it with the normalized data, featured data, and all data that you have at every step, parsing, filtering, and normalization. So here I will create different runs with different methods. So first one I use lemma, then I will use dseq2 on the featured data, then I will use scde. So CDE is actually very long to run, but once it's finished, then you can see actually two boxes. The genes that are found down-regulated in the first selection, which was CRD, and the ones that are upregulated in the first selection. So here I'm interested more into the genes that are upregulated in CRD cells. So we click on this box, and I see a list of the genes that are found as upregulated. So here, for example, we can see ACTC1, MyoZ2, and those are genes that are known to be upregulated in cardiac cells. And if you go back, then now we can see, okay, we have differential expressed genes. What are they corresponding to, biologically speaking? So we can do a functional enrichment analysis. So first you need to select the differential expression results, because the enrichment analysis is only based on a list of genes that are found at the D step. And then you select which of the D genes you want to enrich, whether the up-regulated or down-regulated ones. Here I selected the 100 top differential expressed genes, up-regulated. And then you select the database of gene sets that you want to use, Gene Atlas, Go, or KEGG. And you run it, and it performs a Fisher's exact test and gives you the results. So here I run all my enrichment on the lemma DE results. And then, so you see I tried different database. And if we focus on the KEGG, for example, results, and you click on the result page, and you see that there is only one pathway that is found as deregulated, which is dilated cardiomyopathy, which makes sense because it's cardiac cells. And if you go back and look at the Gene Atlas results, so you see that the first hit is actually heart, which makes sense again because it's heart cells that we are looking at. And similarly with Go results, then you see many pathways and Go sets that are related to cardiac and muscle. So here you see one of the results I told you is the SCTC1 gene. So what I will do is that I will just go back to the visualization pane and I will enter ACTC1 to see the expression in all of my cells. And here you see that it very nicely highlights this cardiac cluster on the bottom left. Then you can put the plot as full screen, and you can also download the PNG if you want to use it later for whatever publication or like just saving it. Now in the visualization tab you also have two other type of plots, the heat map plot and the correlation plot, and very soon there will be also the trajectory analysis based on monocle. So correlation is very easy, so you just select two cells from your data and it will just show you the correlation between the two cells based on normalized data or featured data, so you select the data you want to see. There is also the heat map feature that allows you to generate a heat map based on gene sets, like from pathway, for example, from KEGG, which we call global gene sets. If you want to see the genes that are in these pathways. But you can also generate the heat map based on the results of a DE. For example, here I select the SCDE results of the upregulated genes and I generate the heat map. So in the web browser, you can actually see the heat map, but it's very big, so it was on purpose because we wanted to be able to see all the different genes. But if you want to have more like a summary of your heat map, then you can download it as a PDF, whether large, so you will see everything, or small, then you will just see the summary. So here I can also go back to the visualization and create my manual selections of cells based on the visualization plots. So if I see a cluster of interest, I can just select the cells of the cluster, and then I can characterize the marker genes just by doing a DE between this cluster and another one, for example, or compared to all the different other cells. Here I just wanted to show the ability also of ASAP to do a DE between two selections. So here I created this new selection, and now I want to have a DE between the BATs, so the brown adipocytes, and the cardiac cells. So here you see again the FABP4 gene, which is very well known to be upregulated in brown adipocytes. That stands out. And again, if you go back to the visualization and you plot it, then you see it very nicely, highly expressed in this BAT cluster. Thank you.